Hello, my name is Rohma and I will be discussing my research project in the following presentation. For my research project, I used computational chemistry approaches to discover drug-like ligands for COP1 that could be used to make small molecule inhibitors or PROTACs. I conducted this project under the guidance of Dr. Shapira and the University of Toronto Department of Pharmacology and Toxicology. My protein of interest for the project is an E3 ubiquitin ligase by the name of human constitutive photomorphogenic 1, also known as COP1. COP1 was first discovered in thale cress, Arabidopsis thaliana, but it has also been discovered in mammals, birds, and amphibians. It is encoded by the COP1 gene and found on chromosome 1q24.1 in humans. In plants, it is engaged in light-mediated development, and in humans, it functions both as a tumor promoter and a tumor suppressor because it interacts with proteins involved in cell survival, growth, and development. Overall, as an E3 ubiquitin protein ligase, COP1 mediates the ubiquitination of target proteins, leading to their proteasomal degradation. And it has many downstream targets, including the Jun proteins, p53, and the CCAAT enhancer binding protein alpha. As an E3 ubiquitin ligase, COP1 is a member of the evolutionary conserved ubiquitin proteasomal system, where it facilitates the polyubiquitination of target proteins. Polyubiquitination occurs in a multi-step process that starts with the E1 ubiquitin activating enzyme activating ubiquitin with ATP, which then transfers ubiquitin to the E2 ubiquitin conjugating enzyme. And lastly, the E3 ubiquitin ligase and the E2 enzyme work together to transfer ubiquitin to the substrate. Once the target protein is polyubiquitinated, it is then recognized by the 28S proteasome complex, which eventually leads to proteasomal degradation. COP1 is made of three conserved domains: an N-terminal ring finger domain, an internal coiled domain, and a C-terminal 7WD40 repeat domain. Each domain is involved in protein-protein interactions. The ring finger domain has a conserved sequence of cysteine and histidine residues that form a cross brace with two zinc atoms, and its function is to aid in the recruitment of an E2 ubiquitin conjugating enzyme to transfer ubiquitin to target proteins. The central coiled domain promotes homodimerization and heterodimerization of COP1 with other proteins. And finally, the C-terminal WD40 repeat domain is the region of interest since it facilitates COP1's interaction with its substrates, as it houses the substrate binding pocket of COP1. The WD40 domain is made up of 4 antiparallel beta strands that fold into a propeller-like structure known as a 7-bladed beta propeller. The beta propeller is stiff because there are hydrophobic interactions between the beta sheets. In human COP1, the beta propeller is folded and stabilized by an N-terminal helix made up of residues 390 to 401, which is coupled to the propeller's 6th blade by a salt bridge connecting lysine and aspartic acid. Additionally, the beta propeller has a 15-residue loop with numerous acidic residues. The top face of the beta propeller interacts with target motifs, whereas the bottom face is responsible for interacting with other E3 ligase complex components. As stated previously, COP1 operates both as a tumor promoter and a tumor suppressor. It has been shown to target tumor suppressors like p53 and oncogenes like Jun for degradation. COP1 plays an oncogenic function in cancer and has been overexpressed in a variety of malignancies, including breast cancer, ovarian cancer, and pancreatic cancer. As a tumor suppressor, the loss of COP1 expression has been seen in melanomas, ductal breast cancers, and gliomas. So it can be said that COP1 has many functions, but For the research project, I focused on one of its oncogenic functions. The function of interest in this research project was the impact of COP1 on CEBP alpha. The CCAAT enhancer binding protein alpha, also known as CEBP alpha, is a tumor suppressor that controls myeloid cell differentiation and has been linked to the development of leukemia. Mutations in CEBP alpha are seen in 7 to 9% of acute myeloid leukemias. COP1 serves as an E3 ubiquitin ligase for CEBP alpha, inhibiting its activity and preventing immature myeloid cells from differentiating, ultimately leading to the onset of cancer. The action of COP1 on CEBP alpha in this case shows its carcinogenic functions. COP1 interacts with a mediator in the process known as TRIB1 rather than attaching directly to CEBP alpha. TRIB1 is a member of the triple peptides family of proteins and is required for the COP1-mediated degradation of CEBP alpha. In human AML, abnormal expression of TRIB1 has been discovered, and TRIB1 contains a C-terminal COP1 binding motif which allows it to interact with COP1. Previous research on the interaction between COP1 and TRIB1 has revealed that the substrate recognition site of COP1 has a shallow binding pocket and can recognize the bound peptide TRIB1's consensus valine to proline sequence. TRIB1 residues D355 forms a salt bridge with residue K472, and the top face of the beta propeller of COP1 engages in peptide binding, whereas the sides and the bottom face interact with other E3 ligase complex components. The top face of the WD-40 beta propeller also has positively charged residues that exert favorable electrostatic interactions with the consensus motif on TRIB1. According to certain studies, the TRIB1 binding site is shared by other COP1 protein substrates, and peptide phosphorylation affects the binding affinity and competition for COP1 binding. Once TRIB1 is bound to COP1, CEBP alpha is brought into close proximity of COP1. COP1 can then polyubiquitinate CEBP alpha, leading to its subsequent proteasomal degradation. This whole process and the action of COP1 inhibits myeloid differentiation and can lead to the onset of leukemia. Overall, many essential residues are involved in the COP1-TRIB1 interaction. However, the complete interaction is not understood, so the structural analysis undertaken in this study will allow a better understanding of COP1's interaction with TRIB1 and CEBP alpha. The purpose of this research effort is to choose drug-like ligands that bind to the druggable target site of COP1 using innovative virtual screening methods. These drug-like ligands can then subsequently be used to create proteolysis targeting chimeras or PROTACs and small molecule inhibitors for COP1. PROTACs are two molecules bound together by a linker to form a two-domain overall structure. One domain of the PROTAC binds to an E3 ubiquitin ligase, and the other binds to the target protein. Once bound, the PROTAC allows the target protein to come into close proximity to an E3 ubiquitin ligase, which can then polyubiquitinate the target protein, leading to its subsequent proteasomal degradation. For the purpose of this study, drug-like ligands can be used as PROTACs to target and degrade COP1. And possibly inhibits its potential oncogenic effects in leukemia. PROTACs are also catalytic and can be regenerated after use. For the research project, ICM Pro, a computational chemistry software that is produced by MalSoft, was used. At first, ICM Pro was used to investigate the structural and functional properties of COP1. A visual inspection of the WD-40 domain of COP1 was performed using the PDB code 5HQG of human COP1. The interaction between COP1 and its substrate TRIB1 was also visualized. Crucial residues that facilitate the COP1 TRIB1 interaction were then identified through structural analysis. The screening strategies were divided into a receptor-based and a ligand-based virtual screen. In the receptor-based virtual screen, the WD-40 domain of COP1 served as a receptor to which a massive library of fragments was docked. The WD-40 domain structure of plant COP1 and its substrate HY5 with the PDB code 6QTO was then used for fragment docking. The plant-specific PDB code was used for the next procedures since it allowed for a higher resolution into all areas of the protein structure. Receptor-based virtual screening yielded a list of distinct fragment hits that were able to interact with COP1. The fragments from the docking output were then assessed for hydrogen bonding and hydrophobic interactions with COP1 in an interaction analysis. Hydrophobic interactions were determined by measuring the distance between COP1 and fragment residues of 3.5 angstroms or less, and hydrogen bonds were visualized with the hydrogen bond option on ICM Pro. Next, the ligand-based virtual screen was applied. Using the endogenous ligand of COP1, HY5, a modified template was first produced. The template was made by removing unwanted residues from the endogenous ligand, and the backbone carbon atom occupancy was adjusted to 0. This template was then employed as the input ligand for the high-throughput virtual screen called the Rapid Isosteer Discovery Engine, or RIDE search. RIDE is an atomic property field-based chemical similarity search engine. It looks for similarities between atomic property fields of the template and prospective drug-like ligands from a large chemical library. Hydrogen bond donors, hydrogen bond acceptors, lipophilic residues, charged residues, hydrophobicity, and other characteristics are all analyzed by atomic property fields. A RIDE search was first performed in a small compound library, but this was then followed by a larger RIDE search using a very large compound library that then yielded many drug-like ligands. The drug-like ligands received as an output of the RIDE search were subsequently docked to the COP1 receptor pocket for an integrative analysis. And finally, the top 3 drug-like ligands were chosen based on the greatest RIDE score, the lowest but most favorable docking score, and the best hydrophobic interactions. Now I will outline the results that were obtained throughout this study, starting with the structural analysis of COP1 and its substrate. Initially, the 7-bladed beta propeller structure of the WD-40 domain of COP1 was seen in ICM Pro, and the structure of the TRIB1 peptide attached to the WD-40 domain of COP1 was visualized. It was seen that TRIB1 binds to the top face of the WD-40 domain of COP1. Following the overall structural examination, a complete picture of the TRIB1 peptide attached to the substrate binding pocket of COP1 was created and evaluated. The interaction between TRIB1 and COP1 was then visually inspected to detect key peptide interacting residues. These residues were outlined based on their proximity to hydrogen bonding groups on the peptide. The key residues were threonine 618, phenylalanine 645, cysteine 559, and tryptophan 517. Now I will outline the results that were obtained from the receptor-based virtual screen. In this section, computer docking experiments were performed to produce a hit list of fragments that would interact with key residues of COP1. From the output, around 100 fragments were visually assessed for common COP1 interaction hotspots. A hydrogen bonding analysis revealed that 31.4% of the 443 fragments formed a hydrogen bond with the nitrogen of F595. 26% of the fragments formed a hydrogen bond with the hydroxyl group of tyrosine 441. 21.2% formed a hydrogen bond with the oxygen of a water molecule, and 17% formed a hydrogen bond with the oxygen of threonine 568, and finally, on serine 375, 10.2% of the fragments formed hydrogen bonds. For the hydrophobic interaction analysis, it was found that out of 3,086 segments, 70.9% had a hydrophobic interaction with phenylalanine 595. A distance of 3.5 angstroms or less was used to evaluate hydrophobic interactions. On phenylalanine 595, 66% of the fragments formed a hydrophobic interaction. A hydrophobic interaction with a carbon on tyrosine 554 was generated by 62.3% of the fragments, and 60.6% of the fragments developed a hydrophobic interaction on another carbon of F595. And finally, 14.9% created a hydrophobic interaction on site F595CZ. Some structural representations of the docking output fragments that interacted with COP1 were also created. The hydrogen bonds and hydrophobic interactions between the major interaction residues of COP1 and the fragment hits were shown. Here, we can see one of the many fragments forming hydrogen bonds with the crucial COP1 residues F595 and Y441. This image depicts the same fragment but in a mesh view. The fragment is seen inserted inside the binding pocket of COP1 while interacting with the key residues. Hydrophobic interactions were classified based on the distance between the fragment and the COP1 residue atoms, with anything less than 3.5 angstroms being termed a hydrophobic interaction. In this image, we can see a fragment creating a hydrophobic interaction with residue F595 of COP1. The fragment is in a pi-pi interaction with the residue, and the distance between the atoms is roughly 3.5 angstroms. In the ligand-based virtual screen, the endogenous ligand HY5, a transcription factor that binds to plant COP1, was used to create a template for the RIDE search and find drug-like ligands for COP1. The RIDE search template can be seen superimposed onto the endogenous ligand for plant COP1, HY5, with certain atoms of the template, namely H10, N1, C6, and C7, adjusted to an occupancy of 0. Both the RIDE template and the HY5 ligand are inserted into the substrate binding site of COP1. The RIDE search resulted in a list of several drug-like ligands that were similar to the RIDE template, and the output of the RIDE search was then docked against COP1. A scatter plot was constructed to compare the RIDE score and the docking score of the drug-like ligands. The first drug-like ligand, designated as hit number 1, was chosen based on the highest and most favorable RIDE score. The second drug-like ligand, denoted as hit number 2, was chosen based on the lowest and most favorable docking score, and the final drug-like ligand was chosen after a detailed visual inspection of the most favorable drug-like ligand, establishing hydrophobic interactions with critical COP1 residues. The visual representations of each ligand hit can be seen. Hit number 1 was chosen based on the highest and most favorable RIDE score. Hit number 1 can be seen forming a hydrogen bond with a critical COP1 residue, F595. The hit can then be seen overlaid onto the RIDE template and placed favorably into the substrate binding pocket of COP1. The CPK image of the ligand hit was also generated to check for possible receptor conflicts, and none were found. Hit number 2 was demonstrated to form hydrogen bonds with COP1's F595 and Y441 residues. This hit was also well superimposed with the RIDE template and inserted favorably into the substrate binding pocket. There was also no conflicts between the hit ligand and the receptor as seen in the CPK view. Hit number 3 was seen to generate the most ideal hydrophobic interactions with the critical COP1 residue F595. An atomic distance of 3.53 and 3.36 angstroms was used to depict this hydrophobic interaction. Hit number 3 was placed in the substrate binding pocket of COP1 and overlaid onto the ride template, much like the preceding hits. There were also no receptor conflicts in the CPK view for hit number 3. Overall, the top 3 hits had good ride and docking scores, as well as favorable hydrophobic and hydrogen bonding interactions with COP1's critical residues. Based on the final research results, the top 3 final hits are the most promising drug-like ligand candidates for this research and can be employed in future studies. It was seen that the hits established hydrogen bonds with critical COP1 residues such as F595 and Y441. The capacity of a drug candidate to form hydrogen bonds is important criteria for determining whether it will have a favorable interaction with the receptor. Hydrogen bonds are necessary for drug receptor binding because they ensure that the substrate remains stable after it is attached to the receptor. Furthermore, strong hydrogen bonds enhance geometric alignment of a substrate to its receptor. Hydrogen bonds between the ligand and the receptor also increase the ligand's affinity because it makes it more favorable for hydrogen bonding and can result in the displacement of protein-bound water molecules into the solvent. This can then reduce the amount of interference produced by protein-bound water molecules. Hydrophobic interactions are also important factors of receptor-substrate binding. Because they are the fundamental driving forces of receptor-substrate interactions and are very important considerations in drug development. The hydrophobic effect occurs when hydrophobic forces cause 2 molecules to clump together and bond, reducing the substrate area that would otherwise meet polar water molecules, resulting in fall in entropy, making the interaction more energetically favorable. The hydrophobic interactions in hit number 3 make a good candidate for further research. There are also cyclic residues in the top hit candidate structures which contribute to hydrophobic interactions. The ligand becomes stiffer when it contains cyclic residues, which improves its binding to the substrate pocket. Furthermore, cyclic ligands have less entropy and are more favorable to binding to receptors. Cyclic residues also reduce the compound's flexibility, making it less vulnerable to pre-acting hydrolysis by endopeptidases that favor more flexible ligands. Finally, cyclic residues can also help create pi-pi interactions between the receptor and the substrate, and pi-pi stacking can improve the conformational stability of drug-like ligands, which improves their binding affinity. Another benefit of pi-pi stacking is that it can be manipulated to load drugs into delivery systems. Pi-pi stacking interactions are affected by pH and redox potential in the environment, so scientists can induce or diminish pi-pi stacking and regulate the release of a drug into a biological system. The top 3 hit compounds are also ideally superimposed into the ride template, and a hit compound's structurally similarity to the template determines how likely it is able to bind to COP1, so the top hits are likely to bind. Finally, it was also seen that none of the top 3 hits clashed with the receptor, which is advantageous because receptor clashes would make a prospective drug molecule less appealing. Overall, this study examined the features of COP1 TRIB1 binding and the crucial residues that facilitate this receptor-substrate interaction were identified as F595, Y441, T568, and Y554. The top 3 drug-like ligand candidates were identified by an interactive analysis that considered ride scores, docking scores, hydrogen bonding, and hydrophobic interaction. The ability of these top hits to form important hydrogen bonds and hydrophobic interactions with COP1, their optimal superimposition with the template, and the lack of receptor conflicts altogether make the 3 hits promising candidates for small molecule inhibitors or PROTACs. It is crucial to note, however, that understanding hydrogen bonding and hydrophobic interactions in biological systems is relatively difficult, and other variables may influence drug receptor affinity. As a result, Additional investigations like in vitro experiments to confirm whether these top hits impede the activity of COP1 are required. Future experiments can also employ additional modified ride templates that focus on specific regions of the COP1 binding pocket, such as the deeper binding pocket. So, all in all, the top 3 hits identified in this analysis appear to be very promising candidates for small molecule inhibitors or PROTACs, but more research is still needed to confirm their suitability for COP1. This research project was supported by Dr. Shapira and the University of Toronto's Department of Pharmacology and Toxicology.